Well, thank you, Alex, and the organisers for giving me the opportunity to give you a little snapshot on some of the work we've been doing trying to make a difference in terms of contribution to designing novel anti-epileptic drugs. I'll apologise in advance because it's extremely nascent, certainly compared to the previous two talks that you've heard this morning, but it's getting somewhere. It's getting somewhere. And that's mainly thanks to us being able to hook up with a couple of concerns, IBM at the Thomas Watson Research Centre, which you've probably heard of, and our main partner here, Eisai Inc., or Eisai PLC in this country, which is a very forward-thinking drug company. We're interested in epilepsy for a number of reasons. I have a personal interest in it because that's how I started in science in the first place, but that's not an excuse. The main reason is it's actually quite disturbingly common neurological disorder, and while it doesn't lead to death in many cases, although sometimes it does have a really detrimental effect on quality of life. It affects people's ability to drive, for example, so it affects whether they can be independent or not. It affects their social confidence, so they become rather withdrawn, and it affects the sort of job they can do as well, to really quite severe effects. There's a very famous teaching aid that was released by the Epilepsy Society, which was a video interviewing a number of patients with various different types of epilepsy and actually capturing some of the seizures for training medical students. And there was this one poor chap who had quite a severe temporal lobe epilepsy. It had only really manifest in his mid to late 20s. And it was really, really badly affecting his life. And it actually showed you a seizure, which was really quite disturbing the first time I saw it. But it wasn't until the end of his interview that they revealed that he was, before the epilepsy, a roofer. And he had to give that up after he fell off a few too many times and broke too many bones. So it really can make a difference to how your life pans out. In most cases, it's a chronic disorder. So you don't just get it and then it disappears, you live with it. So the drug companies, to be rather cynical, love it because it means repeat prescriptions. And there are a lot of them. So I think currently there's about 30 drugs available for clinicians to prescribe to people who present with some or other form of epilepsy. And even more if you add on the adjuncts as well. And that's good because you can, if you have the time, present a patient with a drug, tell them to go away and try it. Does it make a difference or does it do nothing at all? Can you handle the side effects? If it doesn't make a difference or the side effects are too horrible, you move them on to another one, etc., until they become stable and content with that pharmacotherapy. But it doesn't always work like that. And one type of epilepsy in particular is particularly troublesome. It's called absence epilepsy. It's troublesome because drugs don't either not work or work for that particular form of epilepsy. Some drugs, which will work brilliantly for one patient, will actually make another patient considerably worse. So you're doing harm by prescribing a drug. And you have no idea of knowing, or almost no idea of knowing, whether the drug that you prescribe is actually going to cause harm or not until it does. So there's a real need to try and stratify this patient group and try and work out why some perfectly good anti-epileptic drugs for certain people are actually having a detrimental effect for others. It's quite a common disorder, and this thing stopped working. No, it hasn't. There we go. It's quite a common disorder. What makes it more disturbing, really, because it's not a severe epilepsy in terms of the motor side effects, is the highest prevalence is in kids, in those formative years from when you're born till about mid-teens. In some cases, it will then just disappear. In most cases, unfortunately, it will then progress onto a different type of epilepsy and mean that you have to try a different anti-epileptic therapy. In terms of primary pathology, it's associated with a whole load of different syndromes. So getting a handle on what's causing these changes and these differences in different patients at the biochemical level, at the physiological level, is really rather tricky. So getting an evidence-based approach has caused problems over the years and continues to cause problems. Having said that, we are at the state where there are at least 2 identified subgroups of absence epilepsy patients. If you can get them into a hospital for an EEG session, you can usually tell on the basis of the dynamics of the EEG whether they have a typical form of absence or an atypical form of absence on the basis of the frequency of the epileptiform events that occur, just non-invasively recorded via an EEG. That's important if you have the time and money as a primary healthcare professional to get the patient into hospital. The other changes that occur, you find out later, too late, if you like. They differ in levels of loss of consciousness. Some of the people have no loss of consciousness whatsoever. They just go blank. So if you were teaching one of these kids at school, you'd just think that they had a tendency to daydream, which is extremely detrimental for their learning pathway, obviously, because you don't get a diagnosis, and they continue to have these absence events during classes on a daily basis and do not pick up on the same amount of facts that their peers would do. It's also rather detrimental because an awful lot of these epileptiform events occur while the patient is fast asleep. So there's absolutely no sign whatsoever what's going on. But that's where the cognitive deficits come in, because it massively interferes with this consolidation process that we all need to form long-term memories, particularly while we're at school age. And you tend to see changes in their academic performance occurring rather insidiously and slowly, and in some cases, but not all, that can actually detrimentally affect their ability to pick up and use language. So again, further isolating the patient from their social environment. And as I mentioned, one of the primary generators for this is that they differ in their response to anti-epileptic drugs. And for example, ethosuximide, which has been around for ages and ages and ages, is really, really helpful for typical absences, but it makes atypical absences worse. There are a bunch of others as well. Carbamazepine makes things much, much worse in terms of atypical absence. And phenytoin, which is definitely a little bit out of favour at the moment, it's been around for an awfully long time, really does do serious damage if you were to give it to a patient with atypical absences. So we've always wanted, or I personally have always wanted to try and make a clinical difference with the basic science, which is all I can do. In fact, if I ever get there, that will be a sort of Barack Obama drop the microphone moment. That will be me done. I will be a happy boy. It hasn't happened yet, but it hasn't stopped me trying. So we've been trying with the aid of IBM, particularly a chap called Roger Traub, to try and design in silico models based on in vitro models of what we think very nicely represent the different electrophysiological patterns that you associate with the different types of atypical absences. Atypical here and typical here. And what we're finding out is that they actually have different origins in terms of the anatomy in the brain. So atypical absences are generated by processes that are going on in the superficial layers of your neocortex, the stuff just below the skull, whereas the typical absences appear to be generated almost exclusively, although the activity gets projected, almost exclusively in these deeper structures, the thalamus. So as we can pick apart which biochemical, electrophysiological processes at the single neuron level and the network level are going on in those situations, we can start to develop models that go way above and beyond the sort of standard single cell culture-based models, which really don't work with epilepsy very well because it's manifest at the network level. It's manifest at the way different cells communicate with each other rather than what an individual cell wants to do. And just have to throw a bit of data in because I'm a scientist. This is what we have so far in terms of validation of the models that we're generating in collaboration with IBM and ESI. And I've shown just 4 examples of 4 different, very commonly used anti-epileptic drugs here on the atypical. These are the neocortically generated absence seizures. And you can see ethosuximide makes things worse a little bit. Valproate does not work at all. But both of those drugs work beautifully for typical absences. Rufinamide, which is an Eisai drug, or it was originally a Novartis drug, it's now an Eisai drug, works really well if you get the dose right. If you get the dose wrong, it will make things worse. And levetiracetam, this absolutely amazing magic bullet that UCB Pharma came up with, works beautifully. And then on the other side of the slide here, I'm just showing you for one drug, the ethosuximide, some data from the models that we have so far showing the exact same profile that you would see in the clinic, where in atypical absences things get a bit worse, and in typical absences things get very, very much better. So the model's been reasonably well validated. We have tested some novel compounds on it, but unfortunately the data's not of sufficient quality yet to show you. One thing we have worked out, though, which we're hoping to capitalize upon in this drug discovery process, is we've actually, or we think, we've worked out why some drugs make things worse. If I'd have plotted phenytoin, for example, it would have just gone up through the ceiling there. And some things make things better. And it all seems to boil down to whether the drug is able to block a particular type of sodium channel-mediated conductance in certain neurons. And that might not mean anything to many of you, but in terms of drug companies, that's great because that's an identifiable target, and when they are screening their drugs, then they know any activity at that particular target site means they cannot progress that drug on as a potential anti-epileptic drug for atypical absences. So we've learned quite a bit so far, although we haven't got to that goal of actually making a genuine difference. But in the process, we have worked out how you can refine the EEG process if you can actually afford to get a patient into an MEG machine in terms of diagnosing with clinical neuroimaging, because it depends very much on where the activity is coming from, what type of primary pathology, and thus typical or atypical absence, you're actually dealing with. And things have progressed so much in this field at the moment that actually now MEG machines can actually, or if you analyze the data carefully, can actually tell the difference between activity that's generated in superficial and deep cortex. So you should, in theory, be able, from what we have understood from our models, be able to delineate absolutely whether this patient is presenting with typical or atypical absences. It's helped us a lot and our colleagues because we're getting to understand an awful lot more about what's going wrong to get these symptoms in the first place. Eisai are happy so far, anyway, because they see us on the way to actually reducing drug development failure. They really want more drugs out there from themselves in this particular niche market. And if we can identify things that they need to avoid in terms of the binding characteristics of their potential drugs, then that would be good. If we can refine or continue to refine with IBM the in silico models, then we're moving towards the sort of holy grail for pharma because it's so quick and it's so cheap of in silico drug design. But we're quite away from that at the moment. And obviously, the main beneficiary, if we can actually pull this off, are the patients themselves. And I'll just finish with just a few things that I have picked up on the way. This is an absolute, it sounds like a bit of an elk theory, really. Needs to be a mutual benefit. It's actually really hard to get that. I have tried to work with drug companies for over a decade towards this particular goal, not specifically this type of epilepsy, but epilepsy in general. And it's hard. It's really hard. What tends to happen is you misinterpret, and they misinterpret, exactly what you have to offer and what you can deliver. And these collaborations start off, everybody shaking hands, and it's great, and they tend to just drift apart. And it's quite sad, and it's very difficult, but we're getting better at it. One of the problems as a basic scientist we have is the IP protection issue. And mainly as regards protecting the people who actually do the work on this project, the more junior research staff. testing drug after drug after drug is not intellectually challenging. It doesn't make for a happy thesis, and it doesn't make for a happy postdoc or a PhD student. And also, you get issues with some partners saying that, actually, can you just not publish that for another year or so? Just want to check what's going on here. And then there are other sides to it as well, particularly with pharma connections, because they often do not let you play with their key parent compound. They'll give you compounds which are similar, but not the one that they are thinking they might want to get into clinic, just in case you find something that might be a little inconvenient for them. And just, it's the way it goes, and I can see why they do it, but it makes our job extremely difficult, because we're usually dealing with things that are rubbish, actually, in most cases. But there are lots and lots of pluses. For our collaboration with Eisai, we have access to an enormous compound library. So if we get a clue, as I was suggesting we have with this activity at particular types of sodium channel, they have an enormous set of drugs which have been characterized in terms of their binding profiles, that we can say, can we try that one and that one? It's like a child in a sweet shop, really. You could say, some of that, please. And if you hit it, if you hit the gold, and you think, this one could work, are you interested? They have everything available to do all of the toxicology to get this from a laboratory into early phase trials in man. So that is something that you just simply cannot do at the university level. And with the IBM handle, those computational resources and the intellect that drives them don't speed up model generation, massively speed up model generation. And in turn, you know, if we can get that model right, then in theory, we shouldn't need the in vitro animal-based model, and we should be able, on the basis of the binding profile of a drug, be able to just simply say, that one will work, that one will do harm. That's the idea anyway, if we ever get there. So the basic science gets better with a collaboration like this, and the potential pathways to impact become obvious, whereas they're actually just the end of a rainbow, usually when you sit in a lab at a university most of the time. And I'll finish there. Thanks for listening.